All right, we are going to switch gears a little bit. So now we're still in the realm where two genes affect the same trait, but now these things have the same phenotype. And so this is an example that came out of studies in the fruit fly where they isolated a large number of mutations that all had the same phenotype. And so they might be interested, for example, in eye color. So Drosophila eyes are normally red, and they found if they added a chemical mutagen, they could isolate flies that had white eyes. And so the question that they wanted to ask was, how many of these alleles, how many of these mutants had alleles in the same gene? So here we're seeing this example of this done in Drosophila for wings, so whether or not these flies have wings. So they've done this screen. They've taken wild flies that normally have wings, so guys that look like this. They mutated them with a chemical, and then they've isolated a large number of independent animals that all have the same phenotype. So we have these two guys that don't have wings, okay? And then we also have these two guys that don't have wings. So this just illustrates the two different situations that we're interested in. So the genetics that we're interested in. So in the first case, the mutants fall in different genes. So the reason that this fly does not build wings is because it has a loss of function in this gene 1. The reason this fly doesn't have wings is because it has a mutation in gene 2. Case 2 is when the mutations are in the same gene. So they're different mutations, so you see they occur in different positions, but in both cases they've lost gene 1. Both of these flies have mutants in gene 1. So how do you go about determining whether this case 1 is the situation or case 2 is the situation? And so the way that you can do that, and this assumes that they're homologous recessive, this is assuming that the mutant phenotype is recessive. So you'll only be able to follow this logic if you understand that. You create an F1 cross between these two. So this fly is homozygous, so it has to supply one of these copies. This fly is homozygous, so it has to supply one of these copies. And then the same situation is here. So we've created an F1 that has one of the mutants on one chromosome and then the other one on a different homologous chromosome. So what happens in these two cases? So in this case, you have a fly that has one intact copy of gene 1 and one intact copy of gene 2. So assuming the mutants are recessive, that means they have one normal copy of each gene, and so complementation occurs. And so the flies are able to develop the wings just like they're normally supposed to, because they have one good copy of each. In the second case, when the mutant falls within the same gene, in both cases you've knocked out gene 1. And so no complementation occurs. So in this F1 individual, they're not able to build a wing because neither one of the homologous chromosomes is able to supply an intact copy of gene 1. So complementation analysis is that goal. So you might have, let's say you have 30 different mutant flies that don't have wings. And you want to ask how many of these flies are able to, how many different genes do these mutants affect? So is it one gene? Is there only one gene that's necessary for building a wing? Is there 2? Is there 3? Is there 4? And so you can build these complementation groups by asking how many of these genes complement each other. So we'll see a little bit more of that in later activities. All right, the final thing that we are going to cover is sex-linked trait. So sex-linked traits occur when the gene is on the X chromosome. So what if the gene is on the X chromosome? And so why is that a thing? Well, the reason that's a thing is because males only have one copy of the X chromosome. So males are not diploid on the X chromosome. They're haploid on the X chromosome. So if there's a gene that is on the X chromosome, that affects things. And so we're going to see really quick how something like this happens. So how does this happen? Okay, so let's say we isolate mutant flies, and so the mutant flies have red eyes or white eyes. So wild type flies have red eyes, so the mutant flies have white eyes. And let's say this actually occurs on the X chromosome. This gene is on the X chromosome. So we are going to signify this by calling this allele X or XW. So these are wild type, and then these are white eyes. All right, so let's do two crosses, one between males, red females and white males, and then let's do white females and red-eyed males. So we are going to put the genotype underneath the animal. So in the first case you have XX crossed with XWY, and the second one you have XW crossed with XY. So this individual is red, this individual is white. And so you'll notice there's an imbalance. So in the first case, even though it seems like you're doing a red with a white, in the first case there's two copies of the wild type X and one copy of the mutant X within the parents. And then in the other case there's two copies of the mutant X and then one copy of the wild type. So when you do this cross, this animal right here can only produce one type of gamete. Can only produce the X. The male can produce this or this. Okay. So because the sex chromosomes pair during meiosis, they can either only do the X chromosome or the Y chromosome. And so the result of this cross is either this or this. So this would be a red female because it's XX. This would be a red male because it's XY. And so that is a result. You have all red-eyed flies. Whereas in this case, you have a individual, the female has to produce the XW, and then the male can produce either of these. So in this case, you have a female that has red eyes, but you have a male that has white eyes because we just followed out the genetics. And that's what this part of it shows. So one thing that's very interesting is that you can have a cross, and if you do the opposite cross, you get completely different results. So in one case, you get 100% red eyes, in the other case, you get half and half. And then the other thing you'll notice is that the phenotypes are actually linked, we call it linked, to the sex of the animal. So the males all have the white eyes, and the females all have the red eyes. And we can also, for example, do a intercross between these, crossing the male and female flies together. And so the result of this is shown right here. And so you can see that all of the females are red, and then half of the males are white and half of the males are red. And then in this cross, we see one half of each of the females are red and white, and one half of the males are red and white. So we've lost the sex linkage, for example, in here. It's just 50-50. Whereas here, we still see that sex linkage. So, you know, a lot of this you just have to be able to follow kind of the process, and if you can follow the process of you know, doing the Punnett square, you can usually work out kind of all of the problems. But you know, it's important just to see what happens when you have genes that are located on the X chromosome. And these also can be sex-linked traits can occur in humans. there are diseases such as color blindness, hemophilia, that are linked to the X chromosome. And so you'll also have to be able to do this by analyzing pedigrees to be able to distinguish it being an autosomal trait. So is the trait on one of the autosomes, or is it a sex-linked trait, meaning it's on one of the X chromosomes? And so, how do you do that? Well, there are certain things there are certain rules that you have to look for. And one of the rules, for example, is if you have a female with the trait that mates to a male without the trait, all of the males must have the trait. So this is an example of kind of like a rule that might be violated. So if this was autosomal, it would look something like this. So in this case, if it was autosomal, none of the individuals would have the trait. None of the offspring would have the trait. Where in this case, because it's sex-linked, we know all of the males of this cross have to have the trait, but none of the females can. So that's an example of something you could look for in a pedigree to determine is this sex-linked or not. All right, so for example, you'll notice this female has the trait and all of her male sons have the trait. So that is an example of one of the rules that I just was mentioning. So again, the best way to kind of get comfortable with these problems is to work through them. So the activities will be useful, I think, for kind of going through all of that.